So for part 3 of this lecture, we continue with the mechanisms of the antimicrobial drugs. In the last section, we were talking about inhibitors that inhibit bacterial enzymes. Similarly, you have inhibitors that can block viral enzymes. And so when you block a viral enzyme, you're blocking some aspect of its ability to reproduce inside of a host cell, because viruses have to enter their host cells. And then what they do is they produce enzymes necessary to make more virus. And usually at the end of the viral infection, or as the viral infection progresses, the new virus are released from that host cell. In some cases, it causes the death of the host cell, but in other cases, the host cell is kept alive just in order to keep producing and releasing new viruses. And so you see here a few of these, amantadine and rimantadine, maybe something you've heard of before, and maybe doing a little online, a little, I would say, informal research on some of these might be something you can do, but you don't need to memorize the names of these. A lot of times with the antivirals, you'll see this VIR somewhere in the name. Not always, but sometimes. So there's a drug called acyclovir, which is an antiviral drug for herpes, treatment of herpes. But in any case, so sometimes you get that little hint in the name, but there are these inhibitors of viral enzymes, just like there are drugs that are inhibitors of bacterial enzymes. We have some drugs that block DNA replication and RNA replication. Certainly a bacterial cell or a virus, as it's reproducing, and even a eukaryotic pathogen, as it's reproducing, it needs to make more DNA and RNA. But the thing is, these kinds of drugs will not only affect the pathogen, they will typically also affect the host cells. They don't have that selective toxicity that we really want. They have some level of selective toxicity, but it's usually not very high. So if you use these drugs, typically you will not only affect the bacterium or the virus, for example, but you will also affect the host cells. So these are typically kind of maybe used in conjunction with some other drugs, but they do tend to cause some toxicity or some serious side effects to the host itself. One that's pretty well known is called AZT. It's a molecule that is very similar to thymine, but when it is incorporated into DNA, it causes, I think it's a strand terminator, but it definitely causes a problem during the DNA synthesis. So as a DNA virus is trying to reproduce, it will have trouble when it incorporates this AZT into the DNA. And so, but also you can have your normal DNA, your human cells, also incorporating this. So it creates problems for any cell that's dividing or reproducing in the body. But it was a first line of defense against some viruses, for example, HIV and hepatitis B. And I think it's still used to some extent, usually more in combination with some other drugs. You have drugs that block something called DNA gyrase, which is an enzyme necessary in DNA replication. And this drug here, the quinolones, act particularly against the prokaryotic version of that enzyme. And then inhibitors of RNA polymerase, that would involve blocking transcription. Rifampicin blocks the bacterial RNA polymerase. So that's the enzyme that would make like mRNA in a bacterial cell. But like I said, these don't usually have the level of selective toxicity that you're hoping for with an ideal drug. And we've already mentioned some antiviral drugs that act as enzyme inhibitors, but on this slide, we're looking at molecules that prevent a virus from attaching and entering the host cell. And so that's a different aspect of the virus life cycle. And when viruses enter a host cell, they have to be able to recognize and attach to certain proteins on the outer surface of that host cell. And so by developing drugs that block those connections, you can block or prevent the virus from being able to attach or enter a host cell. And then there even are some drugs that don't prevent the virus from entering the host cell, but they prevent the virus from a process called uncoating. The virus has to lose its protein coat, which is sometimes called the capsid. It's always called the capsid. And the uncoating process can be blocked by certain drugs. So if you prevent a virus from entering a cell or from uncoating, once it does enter a cell, then you will have found a drug that can prevent the infection. And so there have been some new drugs for HIV that have been that achieve this mechanism. And one that I listed down here, miraviroc, binds to a receptor called CCR5 and blocks it. And once all your CCR5 receptors are blocked, HIV cannot find any receptor to attach to if they're all blocked. And so HIV usually binds to an available CCR5 receptor as a part of its entry process into your cells. But if you have taken a drug that blocks those receptors, then the HIV will not be able to enter your cells. If a virus can't enter your cells, your immune system will eventually clear it out of your body. And because it has not entered your cells, you don't have any infection. So I was mentioning what the ideal drug would be something that's readily available, inexpensive. We know sometimes that's not the case. Chemically stable, so it can stay on the shelf or in a refrigerator, for example. Easily administered, non-toxic, non-allergenic, and then has a good selective toxicity against a wide range of pathogens. But as I mentioned in another chapter, ideal is, in a perfect world, we might be able to have all of these characteristics, but usually or often we have to make some compromises based on what drugs we have discovered. So we are a little bit limited. We don't always have the ideal drug available to us. All right, I'm going to take a break and we will pick up on this in the next lecture segment.